Assalamu alaikum dear students. So we are going to start a new unit today, which you can guess from this slide. It is acid-base balance. So what we are going to study in this unit will be the basic terms and definitions and the brief review of the physiological mechanisms for regulation, including the chemical buffer systems and the renal mechanism and the respiratory mechanism for the regulation of acid base balance. And then we will be briefly doing the classification of acid base imbalances, and then in the end the arterial blood gas interpretation for the acid base disorders. So what is an acid? An acid is a chemical substance which contains hydrogen ions or a hydrogen containing substance which dissociate in the solution to form हाइड्रोजन आयंस. ठीक है. So any ionic or molecular substance which can act as a proton donor is known as an acid. There are two types of acids: strong acids and weak acids. The strong acids are HCl, H2SO4, H3PO4, and the weak acids include carbonic acid, H2CO3, and acetic acid, which is CH3COOH. And then comes the variety of volatile acids and fixed acids. The volatile acids mean those acids which are unstable and quickly evaporate, diffuse in the gaseous form. They are equal, and their amount in the body is equal to 20,000 milliequivalent per day and produced by oxidative metabolism of carbon dioxide, carbohydrate, fat, and protein in the form of carbon dioxide. So, on the average, 15 to 20,000 millimole of carbon dioxide is produced per day, and this carbon dioxide is excreted through lungs. And then comes the fixed acids. The fixed acids, these cannot quickly dissociate and evaporate through the solution in the form of a gas, and hence they are known as the fixed acid. They need kidneys to excrete them or get rid of from the body. Their amount is 1 milliequivalent per kg per day, and they do not leave the solution form once produced. They remain in the body until eliminated by kidneys. Examples include sulfuric acid, phosphoric acid, and organic acids. They are most important fixed acids in the body, and they are generated during catabolism of amino acids. For example, oxidation of sulfhydryl groups of cysteine and methionine, and then phospholipids, hydrolysis, and then nucleic acid metabolism. And from all these volatile and fixed acid, the physiologically important acids include carbonic acid, which is a volatile one, and then the phosphoric acid, which is a fixed acid, and then pyruvic acid and lactic acid. Both of these are the products of glucose metabolism. And then what is a base or an alkali? A base or an alkali is defined as a chemical substance which can accept a proton or hydrogen ion. Any ionic or molecular substance which acts as a proton acceptor, this is known as a base or an alkali. There are strong alkalis, for example, sodium hydroxide and potassium hydroxide, and then there are weak alkalis, for example, sodium bicarbonate, ammonia, and sodium acetate, CH3COONA. And among these, the important physiologically important bases include the bicarbonate ion and the biphosphate ion. So the bases, the weak bases, they are negatively charged. And what is a buffer? Buffer, it is a mixture of an acid base solution or which has an ability to balance changes in pH. So the ability of an acid-base mixture to resist sudden changes in pH is known as buffer. Buffer is a solution of weak acid and its corresponding salt. Buffer resists a change in pH when a small amount of acid or base is added into it. By buffering mechanism, a strong acid or a strong base is replaced by a weaker one. So here you can see the pH scale. The 7 is the neutral pH, that is the middle one, and when we go towards the left side, the pH decreases and the solution becomes more and more acidic. And if you come towards the right side, the pH increases and the solution becomes more and more alkaline. So pH is defined as the negative log to the base 10 of molar concentration of hydrogen ions, and since the molar concentration, the log and of the molar concentration is in negative, so if the hydrogen ion concentration increases, the pH decreases, and when the pH decreases, the solution becomes more acidic, and if the hydrogen ion concentration falls, the solution becomes more basic or alkaline and the pH increases than 7. And what is the normal body pH? The homeostasis of the pH is tightly regulated, and that of extracellular fluid is 7.4, and the normal range of pH in the blood is 7.35 to 7.45. If it falls below 7.35, this is known as acidosis or acidemia. If it rises above 7.4, this is known as alkalosis or alkalemia, and acidosis with pH less than 6.8 is lethal, can lead to death, and alkalosis with pH more than 8 is again lethal and it leads to death. So the blood normal pH from this diagram you can remember 7.35 to 7.45 is a happy normal pH. Below this is acidosis. Above this is again acidosis and death when the pH falls below 6.8, and death when the pH rises above 8. Any small changes in the pH can produce major disturbances in the enzymatic functions because they work only with the narrow pH range, and acid-base balance can also interfere with the electrolytes function like sodium, potassium and chloride, and also affect membrane functions and protein functions, and they can also affect the hormonal actions. And the regulation of acid-base balance is known as the equilibrium between the acid and base elements of the blood and body fluids. The body has three mechanisms to maintain acid-base balance. First is the chemical buffer system, which is the first line and robust acting. The second is the respiratory compensation. It is the second line and it also occurs separately but within from seconds to minutes. And the third one is the renal compensation. This is the third line. However, this is the definitive one. It takes few days, hours or days to settle, but this is the most definitive and accurate correction of any changes in pH. This is also known as metabolic compensation. So here from the Henderson-Hasselbalch equation you can see pH is equal to pKa plus log base over acid, that is negative ion divided by Ha, which is the conjugate base divided by acid. So its metabolic component, that is bicarbonate, is regulated by kidneys, and its respiratory component, which is partial pressure of carbon dioxide, is regulated by lungs. And the first line of defense, as we already discussed, is the chemical buffer system including bicarbonate, phosphate and protein. And the second line of defense is the physiological buffer, from which the respiratory mechanism occurs quickly within seconds to minutes by carbon dioxide excretion regulation. And then this is physiological buffer include the renal mechanism of excretion of hydrogen ion concentration. So the buffer systems are the first line of defense and they are very rapidly acting within the seconds, and buffer system of blood, tissue fluids and cells immediately combine with acid and base to prevent excessive changes in pH, and they do not eliminate hydrogen ion from the body or add them to the body, but only keep them tied up until the balance can be re-established with the help of kidneys. There are three major chemical buffer systems: bicarbonate, phosphate and protein. So here you can see the extracellular fluid and intracellular fluid. This is the classification of buffer system. The intracellular fluid contains the phosphate buffer system, which is present only intracellularly, and the protein buffer system, which include three types, One is the hemoglobin buffer system, which is again intracellular fluid buffer, and it is present only in red blood cells and used to buffer carbon dioxide. The other one is the amino acid buffer system, which is present in all proteins. And the third one is the plasma protein buffer system, which is present extracellularly. So the extracellular fluid contains two type of buffer system. One is a protein buffer system containing plasma proteins and other amino acids, and the other one is the carbonic acid bicarbonate buffer system, which is the major extracellular fluid buffer system. And now we will discuss them one by one. The bicarbonate buffer system is the most important extracellular fluid buffer and also accounts for 65% of buffering capacity of plasma and 40% of buffering capacity of the whole body. And its basic constituent bicarbonate is regulated by kidneys. However, the acidic component carbonic acid is under respiratory regulation. The carbonic acid H2CO3, it dissociates into hydrogen and bicarbonate ions, and the normal concentration of bicarbonate to carbonic acid ratio is 20:1, which means that there is a large reserve of bicarbonate or the alkali in the body as compared to carbonic acid. Here you can see that 20 parts of bicarbonate, which is 24 milliequivalent per liter, is balancing the blood pH on the basis of 1 part of carbonic acid, that is 1.2 millimole per liter. So, what is the mechanism of action of bicarbonate buffer system? If in case a strong acid is added into the blood, this strong acid will release hydrogen ions, which will combine with bicarbonate to form carbonic acid, which is a weaker acid. Hence, there will be slight or only less changes in pH solution. And then if the strong base is added into the blood, that is alkalosis occurs. This base will react with carbonic acid to form sodium bicarbonate, which is again a weak base, and then hence only a very slight increase in pH of the solution will occur. So here you can see that in case of acidosis or addition of extra hydrogen ion, this bicarbonate will split into water and carbon dioxide, and respiratory rate will increase. So more and more carbon dioxide will be released from the lungs out of the body, and hence the partial pressure of carbon dioxide will be reduced in the blood. And also from the kidneys there will be more excretion of hydrogen ions, and the other buffer systems will also accept this hydrogen ion and neutralize them in order to compensate for any fall in pH. And this depends upon the bicarbonate reserve from where bicarbonate ion is coming into the plasma. Then in response to the alkalosis, in case of alkalosis, there is an excess of bicarbonate ion which will be taken up in the bicarbonate reserve, and the hydrogen ion, in case of alkalosis there is decreased hydrogen ion concentration, so the kidneys will generate more hydrogen ion, and the other buffer systems will also release some more hydrogen ion to compensate for any fall in any rise in pH, and also the respiratory rate will be decreased, leading to less excretion of carbon dioxide from the body in the form of respiration. This carbon dioxide will stay within the bloodstream and combine with water to form carbonic acid. Hence the pH will come back to normal or near normal. So, but there are different limitations or certain drawbacks of bicarbonate buffer system. The first of all is this, that it cannot prevent extracellular fluid from changes in pH resulting from increased or decreased level of carbon dioxide, because carbon dioxide is a part of this buffer system and it functions only when respiratory system and respiratory control center. Here you can see that this buffer system depends upon two things, that the bicarbonate reserve is enough and also this respiratory rate and these lungs are functioning properly, and also carbon dioxide is being used as a part of this buffer system. So these three things are the limitation of this bicarbonate system. Then comes the protein buffer system. Plasma proteins in general and hemoglobin specifically are an important buffer system and play an important role in buffering carbon dioxide. There are about 700 grams of hemoglobin in the blood of an adult person, and deoxyhemoglobin is a better buffer than oxyhemoglobin. So the carbon dioxide, when it combines with water, it forms the carbonic acid, which dissociates into hydrogen ion and the bicarbonate. This bicarbonate is exchanged with chloride, and this is known as the chloride shift. This carbon dioxide diffuses across the cell membrane of red blood cell, for which no transport mechanism is required. As carbonic acid dissociates, bicarbonate ions diffuse in the plasma in exchange for chloride ions, which is the chloride shift. Hydrogen ions are buffered by hemoglobin molecules. Hydrogen hemoglobin plus oxygen change to hydrogen hemoglobin oxide, and the only intracellular this the hemoglobin buffer system is the only intracellular buffer system which has an immediate effect on extracellular fluid pH. Any other intracellular buffer system like phosphate buffer system does not have an immediate effect on extracellular fluid. So this hemoglobin buffer system prevents any major changes in pH when the plasma carbon dioxide concentration is rising or falling. Here you can see that this is a red blood cell, and from red blood cell carbon dioxide is being released into the lungs alveoli, and how this is occurring, that bicarbonate is entering within the red blood cell and the chloride is getting released in reverse exchange. This bicarbonate ions, when combined with hydrogen ion, carbonic acid is formed, which quickly dissociates into water and carbon dioxide. So the carbon dioxide can be released to the lungs. And here you can see that when hemoglobin oxide is there, this hemoglobin oxide, it takes up hydrogen ion, so it becomes a hemoglobin hydrogen ion. This hydrogen ion is then regulated over here, and when the hemoglobin is deoxidized, that is, it contains carbon dioxide instead of oxygen, it accepts more carbon dioxide, and this carbon dioxide is also buffered here. Then comes the phosphate buffer system. The phosphate buffer system contains anion hydrogen phosphate, which is a weak acid. It works like the carbonic acid bicarbonate buffer system. However, it is an important intracellular fluid buffer and the renal tubular fluid or the urine buffer system. So the normal ratio of sodium hydrogen phosphate versus the sodium hydrogen phosphoric acid in the plasma is 4:1, and it is kept constant in kidneys. This phosphate buffer system has absolutely no role in extracellular fluid pH, and it accepts sodium hydrogen phosphate accepts a hydrogen ion. So sodium hydrogen biphosphate is formed, and hence this buffer system accepts hydrogen ion. This was all for today. Thank you for being and listening, and please go back home and go through this lecture because this is very important. If you don't go through daily, you are unable to understand. Thank you.